Hey guys, so this video is going to be about wrapping our heads around these levels of eukaryotic gene control. These are the levels at which a eukaryotic gene can be regulated. So what we're seeing here is that this is much more complicated than just simply on or off, like an operon. This is on, this is off, this is turn it up, turn it down. And so the first level, the levels are, as from your last doc, the levels are pre-transcriptional, transcriptional, post-transcriptional, and then post-translational. And those words should make sense to you, right? They tell you when this regulation is happening. So the first one, pre-transcriptional, that is what we learned in our last lesson. So there's really nothing new to say here. Pre-transcriptional gene regulation is whether those histone proteins are tightly wound up, leaving the DNA inaccessible to RNA polymerase to transcribe, right? Or are those histone proteins acetylated? This would be methylated, right? You're not required to memorize that. I'm just reminding you that those methyl groups bind to this. I want you to understand that sort of background concept that small molecules will bind to create changes in this structure. But methyl groups will bind to close the gene off, or acetyl groups will attach to the histone proteins themselves and cause those histones to kind of move apart. And now our gene is on. And you read in the last assignment, hopefully, and did some of those interactives on the University of Utah website with like Lick Your Rat Pups and the GFP glowing protein. You did some of those interactives to show how that might work, and also the concept that environmental factors can lead to this arrangement of a gene or this that could then be actually inherited by offspring. So that's pre-transcriptional. Next level, transcriptional. So this is transcriptional regulation would be involving whether the gene is actually being transcribed or not. And the most important thing to understand here would be transcription factors, right? So if this is my DNA here, this strip, now remember, eukaryotic DNA does not have operons, but there are promoters. So if this is my gene, let's say here, this would be a promoter region, right? And that would be where RNA polymerase would bind. So here is RNA polymerase as a big blobby protein binding. What the most important new thing here is that in order for RNA polymerase to bind to a eukaryotic gene, typically there needs to be one or several transcription factors present. A transcription factor, and for some reason I find that AP Bio students have a tough time with this concept for some reason. Never quite figured out why. So listen carefully to this. A transcription factor is a protein. That's all it is. It binds to either near or at the promoter, allowing RNA polymerase to then bind and transcribe, right? So it might look something like this. Maybe there's transcription factor A, and maybe there's transcription factor, I'll just put them all in the same color, transcription factor B, and transcription factor C. And now this creates a shape on the gene that then RNA polymerase can come in and attach to. And I'm obviously just making up this shape, but what the concept I'm trying to get you to think about in this drawing here is that my picture looks a little bit weird, but like molecules A, B, and C needed to be present for RNA polymerase to transcribe the gene. So you could, now we're starting to get a sense of like how a volume knob might work with genes in eukaryotic cells. Like what if transcription factor A and B were there, but C was not? Maybe the gene is transcribed a little bit, but perhaps RNA polymerase is not as attracted to the promoter region as it would normally be. Maybe when only A is present, the gene doesn't get transcribed at all, even if it's open and accessible, right? So this is the level of transcriptional gene control. The most important takeaway here is this concept of transcription factors, and you'll see some visualizations of these in some textbook images on the doc that I put up today. All right. The next level of gene control is post-transcriptional. Post-transcriptional involves RNA modifications. And you know a little bit about this already, so it's not entirely new. And this is pretty, I would say, hugely important. I'll keep DNA orange, so, or sorry, DNA was orange, so I'll make RNA green. So here is our mRNA that's just been transcribed. We would call this now our pre-mRNA. There's a couple things that are going to happen to this RNA, right? First of all, this is a reminder. We've technically covered this before. A 5' cap is added. 5' cap, right? That's, you don't need to know this, but that's probably I think this is like a modified GTP. If you remember not ATP, but if you remember GTP from some of those cell signaling pathways, this is a molecule. It gets stuck on the front end of it, and then we have a poly A tail, right, attached to the back. That's literally just adenine, 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 A. So that's one of the first things that happens to the mRNA. You could sort of think about both the 5' cap and the poly A tail in the same way, which would be that it stabilizes and protects the RNA, or the mRNA specifically, I should say. So it protects it from enzymatic degradation in the cytoplasm. It makes it actually move a little faster through the cytoplasm. Now, I don't have a lot of expertise in this specific area, but so what I'm saying, right, the next thing I'm about to say could be possibly a little bit wrong, but just to get you thinking about how this could regulate a gene, it's possible that less of a poly A tail maybe makes the mRNA travel a little slower, and a longer poly A tail might stabilize it more and make it travel faster. So it's getting the mRNA out to the ribosome. Is that happening? Is that really turned up all the way? Is it really happening fast? Is it happening a little slower? So that's the addition of the 5' cap and the poly A tail. Now, the other thing in this post-transcriptional level would be RNA splicing. There are spliceosomes. These are complexes of RNA structures that are called ribozymes. Getting a little vocabulary here on you, vocabby I should say, but there's some information on the doc about this. These groups of ribozymes form spliceosomes, and spliceosomes are RNA that chop. They cut out, if you remember, the introns of the gene. So I'm going to do another picture for that, but I just want to pause here. Here's one of these examples where RNA has a catalytic function, right? RNA, not a protein, is cutting up the mRNA strand, right? So what we're going to do is we're going to take out these intervening sequences called introns, and we're going to leave in the sequences that are called exons, right? If you don't remember this, I want you to know this is not new, right? A eukaryotic gene has introns, which are not expressed, and exons, which are expressed, right? So think of the exons as what stays in the mRNA. They get expressed. So I'm going to pop these out. Whoop, there goes one intron. There goes another intron. This would probably happen before the addition of the poly A tail and the 5' cap, honestly. But now, what do we have left? We have this piece has been removed. We have this piece has been removed. We have this piece. So my mature mRNA is only about that long. And let me just stick the 5' cap on again, and the poly A tail. And now we have a mature mRNA strand, right? So notice the mature mRNA strand, the one that actually makes it out to the ribosome, is shorter, right? Introns have been removed. this allows for differential expression of a single gene. This allows one single gene, and this is an important point, to make more than one protein. You know, we knew at the start of the Human Genome Project in the early 90s, we knew that there was about 150,000 to maybe 200,000 proteins in a human being. And so that the assumption was made that it would be about 150,000 to 200,000 genes, but we only found about 20,000 genes. So this is how this was puzzling, and people realized that, oh my God, one gene can actually make more than one protein if you alternatively splice the introns out in different ways, right? So there's some diagrams in the textbook about this that will be helpful, because we could leave it, like for example, we could leave out this exon too. And you'll see a picture visualization of this in the textbook. We're over 10 minutes on this video, so I gotta hurry this up. This is gonna take forever to process and put on YouTube. So the last piece here is translational, or I should say post-translational modification. And this is just, where are we? We're post-translation. The protein's already made, right? So this is just, is the protein on or off? It's just that, you know, all of these factors could have been lined up perfectly. The gene could have been accessible at the histone, the chromatin level, if you will. The transcription factors could have been present for RNA polymerase to attach. The mRNA might have been modified in the right way to get to the ribosome, and the ribosome makes the protein. But if the protein is not phosphorylated or glycosylated or cleaved in a certain way, this is something you should be familiar with, it's not on. So even after all of that, we could still have the gene product be shut off. So just all these different levels of regulation in eukaryotic cells. So here's a, like, let's say inactive protein, and then when I cleave off a section of it, it exposes a binding site, and that activates the protein, right? Or maybe we have our inactive protein like this, and when I attach a phosphate to it, now that opens up a binding site on the protein and it becomes active. So that's post-translational. I'll stop it right there.